Hello class and welcome to lecture 17. Today we're going to focus on the intracellular signaling mechanisms and some of the major pathways. I hope everyone's doing well and let's get started. As we always like to do, we'll begin with a little bit of context. And I guess first a reminder, chapter 16 of your Alberts et al. Text is the assigned reading for today. Now in terms of context, remember that we're in Unit 3, and Unit 3 is all about the proteins, and really specifically protein pathways. So we're thinking about a receiving cell, a cell that's going to communicate with some other cell via typically a ligand, and we're talking about the protein networks that exist inside that receiving cell that allow for signal transduction to work. Now, the whole point of this intercellular signaling and communication is extracellular ligands will have some effect on the target cell. And the effect could be one or multiples of many different things. It could be a signal for growth, a signal for differentiation or a shape change. It could be a signal to divide. It could be a signal to go into stasis or even to apoptose or die. So it's all about the communication and it's all about the intracellular protein networks that get the communication going. That's really what we're focused on for this unit. In my last lecture, lecture 16, I focused on major types of ligands and receptors that get the whole game going. Today we're focusing on the intracellular pathways. Before we go through the outline, let's take a look at this kind of ridiculous cartoon. My goodness, that is complicated. And of course, the point of this is not to overwhelm or even to suggest that you commit to memory any of this. I'm really just trying to illustrate the point that, wow, there's some complicated stuff going on here. So take a look with me at what this cartoon is trying to illustrate. So we're looking here at the cell surface. Obviously the plasma membrane is indicated. This would be the extracellular space and this would be the cytosol. And look at these networks of proteins that exist here. And believe me, this is only a sliver of what is really there, and we're really looking at a tiny, tiny little piece of this larger signaling mechanism that a cell can go through. But let's see what the major sort of salient points are. First, many of these, if not all of them, are set off by an extracellular signaling factor. These are the ligands that are in the extracellular space binding to a receptor, inducing a primary transduction event that then allows tapping into one of these networks, one of these protein networks in the cell. All of these proteins in the network have been already synthesized. So most of signaling is a post-translational event. So the proteins are already made, they're in place, and they're just waiting for activation. So typically it's the ligand that's the prime mover or the primary transducer. I guess really the receptor is the transducer, but what I'm trying to say is it all starts with the ligand. Without the ligand, the receptors don't signal. So the ligand's there, it binds to the receptor, we get a conformational change typically, and we tap into one of the networks. So here's a G protein coupled receptor. Here's the G protein it's coupled to. And now here are some of the downstream targets. Looks like a heterodimer of some sort. One of these then signals into this downstream mediator, which then signals to this downstream mediator, and so on. You can see how the signal just keeps going. It keeps bouncing and bouncing and bouncing. Notice how occasionally there's divergence, where one protein species can activate 1, 2, 3 downstream targets. That's divergence. Okay. And a lot of times these pathways kind of work in parallel, where one pathway, let's see if I can find a good example. Okay, I think I found one. So look at activation of this pathway over here. This is a receptor that's bound to a ligand. It activates this pathway, goes through here, through here, through here, and now to here. And we can trace this one back here, 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 I think. Anyway, my point is sometimes two completely different pathways that exist in parallel converge, and we call this convergence, not surprisingly. So these are sort of interesting aspects of these signaling networks. Sometimes a pathway diverges, sometimes they converge. No matter what, the target is often things like gene expression or proteolysis or modifications in shape by messing around with the cytoskeleton, increased translation, remodeling of chromatin to activate gene expression. You can see myriad effects downstream in these pathways. And remember, by the way, that designation of up and downstream. Anything closer to the start of the signal is upstream. Anything further away is downstream. So, this protein, PI3 kinase, is upstream of this target and this target. And these targets are downstream of PI3 kinase. Okay, so that's a little bit of a introduction and tease. Now let's look at the overview, look at the outline for the class today. So we're going to cover basically 4 major signaling pathways. Okay, first I'm going to give you a little more overview, simplified version of kind of what I just told you, but let's do it one more time just for kicks. Then we'll go into the 4 pathways. First, the cyclic AMP protein kinase A pathway. Second, what we call the PIP2 pathway. Third, what we call the PIP3 pathway. And then fourth, what's known as the ERK-MAP kinase pathway. Part 1 will focus on everything up to PIP2. So this part will be part 1. And then part 2 will come in for the rest. A little bit of a preview for the next lecture, Lecture 18. Lecture 18 is going to focus completely on experimental strategies. Remember, it's not only what we know, it's how we know what we know. So we always need to think about experimental approaches. Instead of introducing the experimental approaches kind of piecemeal, I'm really going to save all of those for one big lecture, and that'll be Lecture 18. So everything will be integrated between 16, 17, and 18 by the next time that we talk to each other. All right, here's the simplified overview. And what I like about this one is it really gives us a sense of how cells don't just respond to a single ligand. They always will express multiple receptor types that respond to multiple different ligands. And oftentimes, cells are bombarded with multiple ligands at every point in time. So let's take a look at the cartoon, and let's imagine that this is our receiving cell. It's the one that's got the receptors, and it's got a receptor for ligand A, ligand B, and ligand C. And so when those ligands are present in the extracellular space and they bind, they initiate a signaling cascade that allows the cell to survive. So these are survival signals that help the cell to be healthy and keep living, essentially. What would happen if we removed those ligands? Well, that's what's shown down here. If we remove those survival factors are no longer suppressing an intrinsic cell death pathway. Weird thing about eukaryotic cells outside of the developmental window, or even sometimes in the developmental window, it just depends on the cell we're talking about. But often for cells, the default pathway is to die, and it's only the presence of those survival ligands that are suppressing these death pathways. Well, if we remove those survival ligands, then we initiate a process called apoptosis. And apoptosis is something we'll talk about next week, but it's the common mechanism of cell death. A common mechanism of cell death, I should say. All right, what happens if we are back up here now and survival factors are present, so A, B, and C are still there, but now we add 2 different factors, factor D and factor E, and we have receptors for those on the cell. Well, now, in addition to obviously surviving by suppressing a cell death pathway, we might get a different signal, like let's divide. So these would be mitogenic signals that cause the cell that receives those signals to undergo mitosis. hence mitogenic. And that kind of cell division is very, very common. This is the kind of thing that goes awry in cancer. And that's one of the reasons, as I mentioned in a previous lecture to you, one of the reasons that we're so interested in these pathways. They have real clinical relevance. Down here, we're still getting the survival factors, so we're happy and alive, but now instead of getting a mitotic signal, we get a differentiation signal. So ligands F and G now are present, and they cause a shape change in the cell. These are just some of the examples, survival, division, morphological change, apoptosis, of effects that ligands and receptors and signaling pathways can have. So along the way, in this unit, we'll be covering a lot of these different mechanisms. All right, now let's begin taking a look at these four major signaling pathways. First, the cyclic AMP protein kinase A pathway. And you can see the nomenclature for this. Cyclic AMP is indicated like that, pKa obviously like that, like an acronym. And the left side of the slide we've already seen. We used this very early in the discussion to sort of introduce the idea of signal transduction, but let's review it, make sure that we're on the same page. So here's our extracellular ligand, epinephrine. It binds to the GPCR, that is its major receptor, and this initiates a signaling cascade involving a G protein. The G protein isn't shown here, but the G alpha is upstream of this adenylate cyclase. And remember the nomenclature here. Some people like adenylyl, some people like me prefer adenylate. They're both the same, two different ways of referring to the same thing. I was trained on this as adenylate cyclase. I cannot think about it any other way. Adenylate cyclase is one of those effector enzymes that takes ATP and converts it to cyclic AMP. So remember the G sub alpha, the G alpha subunit, stimulates the activity of adenylate cyclase, and then it converts ATP to cyclic AMP. And that's what the cartoon is indicating here. So there's a lot in this arrow here. I just unpacked it for you over here. So when cyclic AMP is activated, it then activates its downstream protein kinase A. And let's actually move over to this part of the slide to look at how that activation happens. So protein kinase A initially exists as a tetramer, and we need to remove these inhibitory regulatory subunits before the catalytic subunits can be turned on. Cyclic AMP is what allows for the removal of those inhibitory regulatory units. So there are 4 binding sites for cyclic AMP, and once they are occupied, we change the shape of those blue regulatory subunits, and they pull away from the catalytic subunits. And now we draw that halo of activity, meaning that pKa is on. Now this protein kinase can go and phosphorylate its substrates. So it's not technically accurate the way the artist has drawn it there, what I just circled, but it does drive home the point that cyclic AMP is what drives pKa activity. It just doesn't quite do it exactly the way they show it in the cartoon. This is the realistic way. Suffice it to say, pKa is active, and now it starts activating downstream targets, like phosphorylase kinase. Here's phosphorylase kinase. It's not on yet. Only after PKC phosphorylation is it turned on. And remember the addition to the phosphate group, the phosphate comes from ATP. So what the protein kinase does is it catalyzes the nucleophilic attack from the substrate onto the ATP, the phosphorus within the ATP molecule. And that's what allows the covalent attachment of the phosphate group. So the kinase catalyzes that transfer and that nucleophilic attack. Now that the phosphorylase kinase is active, it can turn on the next enzyme in the cascade, glycogen phosphorylase. Here it is in the inactive state. Here, after phosphorylation, it's active. And then glycogen phosphorylate helps to convert glycogen to glucose for glycolysis. So that helps to give fuel for the cell. Okay? So that's one example of a cytosolic effect Within the cyclic AMP PKA pathway, we drive metabolism in the cytosol and make a lot of glucose available to start driving, the process of metabolism. So let's come over to now this part of the slide, because this helps us to see the idea of divergence in the pathway. Everything on this side is identical to what I showed you over here. Let's just quickly go through it. Activated PKA, activated phosphorylase kinase, activated glycogen phosphorylase, conversion of glycogen to glucose. What the left side of the screen doesn't show you is the divergence, where the same protein kinase A step can also diverge to do something different, like inactivate glycogen synthase. Now, why is that logical and even really helpful in the context of this epinephrine signaling? Well, if epinephrine's goal is to drive glycolysis, and that's certainly one of the things that it does in receiving cells, it would not make sense to build up glycogen, right? It wouldn't make sense to convert glucose into glycogen to store it for later if we really want to increase metabolism in those cells. So let's inactivate the enzyme that builds up glycogen. So that's what I mean by divergence. The same step in the pathway diverges in two different, sort of downstream pathways. And another sort of side note, often we think of phosphorylation as always positively regulating an enzyme activity. That's not always the case. It really depends on the enzyme we're talking about. Here's an example of phosphorylation resulting in inactivation. So remember, it can go both ways with these molecular switches. So continuing with the cyclic AMP PKA pathway, but now instead of looking at effects in the cytosol, we're going to look at effects in the nucleus, because, hey, it turns out this pathway can be involved in a lot of different effects. Okay, previous slide looked at things in the cytosol, altering metabolism. Now we're going to look at how cyclic AMP and PKA can regulate gene expression. So they can play a role in the nucleus as well. Okay, so let's take a look at the role that PKA can play in the nucleus. So here we are in the cytosol, and here's our rise in cyclic AMP. That happens via the activation of some upstream pathway. And here's what we saw earlier, where the cyclic AMP occupies these binding sites on those regulatory subunits. They pull away, and now the PKA is active. Here's what PKA will do now. Instead of having an effect here in the cytosol, it's going to move across the nuclear pore complex to gain access to the nucleus. And there in the nucleus, it's going to phosphorylate a substrate. In this case, a trans factor called CREB. Once activated by phosphorylation, CREB dimerizes. You can see the homodimer indicated here. It exposes DNA binding domains that allow it to interact with a promoter. Okay? And remember, a promoter is a cis element, a DNA sequence that's regulatory and drives gene expression. So the trans factor, the activated trans factor, binds now to the promoter and now drives transcription. It does that by recruiting the RNA polymerase to allow for transcription of a target gene. Okay. So regulated gene expression downstream of a rise in cyclic AMP, and that's why we call this cyclic AMP regulated gene expression. Just a quick word about nomenclature here, CREB and CRE. So CREB stands for Cre binding protein. Okay, that's not so interesting. Cre stands for cyclic AMP response element. So it's a cis element that in a way responds to cyclic AMP. And what we mean by that is cyclic AMP is what gets the cascade going that allows those Cre cis factors to work. So we can't turn on any genes downstream of that Cre promoter unless we have the rise in cyclic AMP. And that's how all of these things get their names. Okay, so transitioning now out of the cyclic AMP PKA pathway and into two pathways that involve phospholipid signaling. And the first is what we call the PIP2 pathway. PIP2. PIP2 is an acronym. PIP2 is a type of phospholipid, and it's a type of phospholipid that's only found in the inner leaflet of the plasma membrane. We don't see PIP2 in the outer leaflet. We only see it in the inner leaflet. And it plays a role in some really important signal transduction pathways. PIP2 stands for phosphatidylinositol 4,5-bisphosphate. And lipids, phospholipids like PIP2 in the inner leaflet, can be cut by lipases. So lipases are like the molecular scissors that cut lipids, just like proteases are the molecular scissors that cut proteins, lipases. So one of the most important lipases is phospholipase C. And phospholipase C comes in multiple subunits, or multiple different flavors rather, and gamma is the one that we're focused on here. So let's take a look at how phospholipase C activation happens and how PIP2 cleavage occurs downstream of it. So here's the extracellular space, here's the cytosol, and here's our ligand, and they're calling this a growth factor ligand. It's going to cause the dimerization of a receptor enzyme. Remember that class of receptor we talked about in the previous lecture? Receptor enzymes contain an extracellular ligand binding domain and an intracellular kinase domain. So they're receptor tyrosine kinases typically. And remember, they exist as these individual monomers. They get dimerized by the ligand, and then there's cross-phosphorylation to activate the kinase portion of the receptor enzyme. And in addition to turning the kinase activity fully on, what the phosphorylation does is it allows for recruitment of SH2 domain containing proteins. So any protein with this SH2 domain can recognize phosphotyrosine and bind. So here's phospholipase C binding via its SH2 domain to a phosphorylated tyrosine on the receptor enzyme. And of course what that does is it puts substrate in proximity to kinase. And you can see how phosphorylation of phospholipase C occurs after binding to the receptor. So now phospholipase C is on, and it's in proximity to its substrate. So the lipase activity kicks on, it now cuts PIP2, and it produces two products. First is diacylglycerol, DAG. Notice that this one stays in the membrane. Second is this one that's soluble, IP3, inositol triphosphate. And there's all of that spelled out. Diacylglycerol for DAG and inositol triphosphate for IP3. And you may guess, and you'd be right, that what these will do is they will activate divergent signaling pathways. Here's a look at those divergent downstream signaling pathways. And let's just begin up here. And we're looking at more of a zoomed in view now of the inner leaflet. And we can see kind of the structure of PIP2. You know, here's the hydrophobic tail, and here's the polar head group, and you can see the phosphate groups on it. That's how it gets its name. Here we're representing phospholipase C cleavage just as a little, indicator over this arrow. The previous slide showed us how it actually gets activated. This one just assumes it's activated. And here are the two cleavage products. Here's the diacylglycerol that stays in the membrane, and here's the IP3. It's basically cutting it in half. So the hydrophobic tail part with this little hydroxyl is diacylglycerol, and then the polar head group is IP3. So what's downstream of these two? Well, hey, check this out. Our good friend protein kinase C, PKC, which you will remember from our discussions in the lab, is downstream of PIP2 diacylglycerol and phospholipase C in this pathway. As a matter of fact, when we were using PMA to stimulate protein kinase C, I didn't tell you at the time because I was saving it for this, but what we were essentially doing was putting in a synthetic version of diacylglycerol. So when we treated those cells with PMA, it was essentially like activating phospholipase C. But we didn't have to activate phospholipase C, and nor did we have to add a ligand and get dimerization of a receptor or any of that other junk. We went right into the middle of the pathway and drove it from the middle by treating with a synthetic diacylglycerol version. So PMA is essentially substituting for diacylglycerol and giving us protein kinase C activation. That's pretty cool. What about IP3? What does it do? Well, let's actually look at this cartoon to get a better look at that. I'll try to blow this up for you a little bit so you can see it better. So let's start here with activated phospholipase C. It's the same kind of downstream information, just rendered a little bit differently and a little more, I guess, stylized. So here's the PIP2 substrate. Now the phospholipase C cleaves it to produce diacylglycerol. Here's diacylglycerol activating PKC. Some isoforms of PKC also require a little bit of calcium to get fully activated. Depends on the isoform we're talking about. What about the IP3? Well, here's IP3. It's soluble, meaning it's not anchored in the membrane anymore. It can diffuse through the cytosol. And you know what it does? It binds to a receptor protein at the surface of the ER, of all places. I bet you didn't know that the ER, in addition to all the things we've talked about previously as part of the secretory pathway and where protein modifications can occur, the ER is also an intracellular store for calcium. Calcium is a really important divalent cation. It's a cofactor for a lot of different enzyme reactions. And it does a lot of things in signaling, as we'll see later. Well, what happens is IP3, when it binds to this little brown receptor, it opens up a channel through which calcium can be released from the ER. And that calcium can do, as the cartoon shows here, it can work as a cofactor for activation of some kinases, or it can do other divergent stuff. So here's a look at the other divergent stuff that calcium can do. In addition to regulating some isoforms of protein kinase C, calcium's also a very important modulator of what we call calcium calmodulin dependent protein kinases. Calcium calmodulin dependent protein kinases. And often we'll just abbreviate this CAMK. So let's take a look at what calcium does here. So here's your cytosolic calcium. It just got released from the ER when IP3 bound to that calcium release channel. Calcium came out of the ER into the cytosol, and once it hits a critical concentration of around half a micromolar, it's of sufficient concentration to do this, binding to a protein called calmodulin. Calmodulin, in the absence of calcium, is shaped kind of like a dumbbell. It's like one of those little 3-pound hand weights that you would use in the gym. So once we add calcium, it binds to these 4 binding sites. And look what it does to calmodulin. It changes the shape dramatically. So conformational change in calmodulin ensues after calcium. Here is the target of calcium calmodulin, which is what we call this complex. This target is the calcium calmodulin dependent protein kinase, or the CAMK. Notice that now it's inactive. But look what happens when the CAM complex wraps around the CAMK. Well, now we expose the active site, we turn on the calcium calmodulin dependent protein kinase. And now this protein kinase can go and phosphorylate a whole bunch of different things, continuing the signal. So what's one of those things? Well, one of those things downstream of CAM kinase is CREB. And you'll remember CREB from the earlier part of this lecture. We saw Krebs as a downstream target of cyclic AMP pKa. pKa could directly phosphorylate Krebs. Well, it turns out activated CAMK can also directly phosphorylate Krebs. That's convergence of two different signaling pathways, the cyclic AMP pKa pathway and the CAMK pathway. And remember, those upstream of CAMK is PIP2. So all that stuff converges on Krebs. And that's one way that we would regulate gene expression downstream of phospholipase C, for example. Okay, so that's the end of part one. And let's just quickly recap what we've seen in part one. So here's the recap. In part one, we looked really at two of the four signaling pathways. We looked at how we could signal through a receptor and various upstream things to eventually get us to a rise in cyclic AMP, which could then activate protein kinase A, and how protein kinase A could go on and initiate a whole bunch of other components of the network. So when we think about this pathway, we're always thinking about these downstream effects. And remember, one of the major downstream effects that we saw was phosphorylation of Krebs and activation of gene expression. We also covered this idea of PIP2 being kind of acted on by some upstream signaling pathways involving also a receptor and a ligand. And then we saw how PIP2 can activate phospholipase C, which can produce diacylglycerol and IP3. And how diacylglycerol is upstream of protein kinase C, and IP3 can mobilize calcium, and that can cause the calcium calmodulin complex to form, and that can activate the CAMK. And what did we see downstream of CAMK? Krebs. So there's your convergence of two totally different signaling pathways. The cyclic AMP pKa pathway up here, the PIP2 pathway down here. Pretty cool. Okay, so the next half of lecture 17 is going to focus on the other two pathways, beginning with the other phospholipid pathway, PIP3. I'll see you in just a couple minutes.